Excellent. Okay. So welcome everyone to the March webinar of PAASTA, which stands for the Paleoproteomics and Archaeology Society for Techniques and Advances. So the purpose of these webinars is to inspire connection and communication between the members of our community. So please feel free to ask our speaker questions at the end of the talk. However, these webinars may contain published and or unpublished data, and resulting conversations and content should not be shared without the express permission of the speaker. So please note that these webinars are not formal presentations necessarily, and the speaker should be contacted directly if you are seeking interviews or statements for reporting. And finally, we want to encourage a safe and positive space for the sharing of knowledge and information in our community. So we ask everyone to abide to our code of conduct, which I will post in the chat. So this week, we are really excited to welcome Ismael Rodriguez Palomo from the University of Cambridge, who will be talking about his article, which is now available in preprint, called Benchmarking the Identification of a Single Degraded Protein to Explore Optimal Search Strategies for Ancient Proteins. So I will pass it over to him.
Okay, thank you, Lindsay, for the presentation and the introduction. Yeah, I'm very happy to present for the PAASTA monthly seminars, and yeah, I'm happy that the community is starting to take off, and so I think this will be a very good point for this presentation, because I think it will be I will be talking about some basic concepts that we've all heard about, that are underlying most of the methods that we use in paleoproteomics, and I will do it through this identification of a single degraded protein that we use as a system, so that we can test different methods and understand better how they use these underlying methods, algorithms, concepts, and so on. So first of all, I would like everyone to remember that in paleoproteomics we tend to use a lot of methods and tools and software that were once created or conceived for the analysis of human or model organisms and cell cultures and tissues, usually in the context of controlled biomedical studies that try to quantify differences between healthy and disease groups, or control versus treatment groups. But first of all, our samples have a lot of uncontrolled variables. They have a history throughout which there were no efforts to be preserved. We are usually more interested in the identification of tissues and or species rather than the quantification, and we usually don't know what's in our sample until we actually analyze the data. Our samples also contain very degraded proteins then. They tend to be a very complex mixture in which contamination plays a very important role, and it's one of the challenges that we face. But even worse, sometimes we deal with extinct species, or at least organisms without a good annotated genome or proteome. So we are actually a very diverse community, even though we all do paleoproteomics. But there's people that look at collagen in bone. There's people that are looking at very complex mixture of food proteins in ceramics. There's people looking at enamel. So we have to consider this and consider the fact that there's also the whole bigger proteomics that look at things in a very different way that we do, but we at the same time share a lot of methods. And so this is just sort of like a cloud of an incomplete cloud of different software tools that are used in proteomics and paleoproteomics that implement very different algorithms and methods. So we basically can divide these tools into the ones that are database search towards the left, middle and the left. And some of them implement what is called open search methods for the discovery of post-translational modifications. And towards the right, we have the de novo tools. And then also some of them, like in the case of PICS or DirectTag, they implement a hybrid method. So they still utilize a database to the search. So these methods rely on algorithms and concepts that for us are crucial to understand, given that we are interested in identifications and that a few wrong identifications can change widely our conclusions. So these main concepts that I will talk about are the target decoy approach, how it is used to estimate the significance of our results. Also, how we deal with post-translational modifications, like, for example, using open search software, and also how de novo tools actually works. We need to understand that there are heuristics integrated into these methods that can bias the analysis in some cases, that they rely on assumptions that if are not met, then we can also get biased results, that sometimes the results are not like deterministic, like we can run the same program different times and maybe get different results. And we need to acknowledge that at the end, there will always be error, mismatches, and false positives. And so we need ways to properly deal with this. But even though this study is a benchmarking in some extent, I don't want it to be like a straight answer to these questions about like which software to use or which parameters, or like what always works and what doesn't. Mostly because there is no right answer to those questions usually, but also because, you know, sometimes good enough is good enough. And if I can answer my research questions with confidence, if I identify enough peptides to back it up, and if the quality of the data is good enough, then, well, I'm also good. You know, I can write my paper, write my thesis, and go home. And then, yeah, maybe afterwards I can worry about, okay, but what if I do this other thing? What happens with things that are not identified? So what this will be more about is, or the intent of this study is to shed some light into these questions, but also probably leave more unanswered questions, which is also good. So what are the caveats of each method and software? Why do I get the results that I get? How can I tailor the parameters that I set to my data, to my context or question, and whether I should actually do it or not? How does the target decoy approach and the FDR estimation work, and how does it influence my result if I use different databases, for example? And how this whole thing interplays with our type of data, like usually complex mixture of degraded ancient proteins with contaminants. And also why the identification rate is so low. Why are there so many unidentified spectra? What are we missing? So in order to look at these questions, we analyzed a progressively degraded beta-lactoglobulin after 0, 4, and 128 days of heating at 95 degrees and pH 7. So this beta-lactoglobulin, or BLG, is a very well known protein milk that a lot of us have seen in our study. It is very persistent, and it's all the time found in different methods, in different substrates, like usually dental calculus or in ceramic. So with this very simple system, what we have is some sort of ground-ish truth. We know what we're looking for in the system, which is this protein, although there might be some other things and contaminants, and because it was purified, there might be other trace proteins. but we know something about it, and also we know the correct sequence of the protein. So then we went on and ran it on the LCM SMS, got the data back. And then we analyzed the data with different software and different parameters to look at the effect, for example, at the effect of the size and scope of the search space and how OpenSearch works and what happens with the accuracy or errors in de novo tools, what would be the coverage of the sequence in the end after after the degradation occurs, and also how this whole thing, like different software, different sets of parameters interplay with the extent of degradation. So we got our data and we did a database search with different narrow and OpenSearch software. We either use triptych, semi-triptych, and non-specific search, and also with two different, very different databases that they vary a lot in size. And then we also run it with a range of de novo tools. And for all this, we set the basic post-translational modifications that most of us usually use, which are the amidation, methionine oxidation, acetylation of the N-terminal, and cysteine carbamidomethylation. Just to keep that there. So before we go into the results, I would like you to think about what is the search space of our analysis, and the search space is the set of peptide ions that we are looking for in our data. In this case, we are testing two different databases, as a very small one targeted for dairy products, and then another one, which is a much bigger one, the whole bovine proteome, and then with three different searches or types of cleavage, like triptych, semi-triptych, and non-specific. And this combination of parameters change the search space. So you can see here how the search space dramatically increases, and this is not even counting peptides with possible modifications. This would be the amount of, let's say, naked peptides. And then, so there would be the search space. And then we have the sample space, which are the ions, the peptide ions that are present in the sample. Then we have what I identified correctly, which is part of the intersection between what there is and what I look for. Then there are things that I identify but are not in the sample. So there would be false positives. Then there are things that I'm looking for that are also in the sample, but I somehow failed to identify. This would be false negatives. The rest of the sample would be what people are calling the dark proteome or orphan spectra, things that I cannot identify. I don't know what they are. The rest of the search space is, let's say, resources that I'm throwing because I'm just looking for these things, increasing the computational time and so on, but I cannot find it. So what I want ideally is for the search space to match as closely as possible to the sample, to limit the false positives and false negatives, and to increase identifications. Let's also talk about, because it's very important and connected to the database, how the false discovery rate works. So in order to have an estimation of how many false positives we have in our results, we use the false discovery rate. But we need to understand that the false discovery rate is just an estimation of the actual false discovery proportion, FDP, which is the actual number, the actual proportion of false positive. And this estimation, FDR, we get it using the target decoy approach. And then we say, okay, under certain assumptions, we expect the FDR shouldn't be too different from the actual FDP. So we get a good estimation. So let's say, for example, roughly, if we have a list of peptide spectrum matches, what I would do is sort them by a given score metric. And then if I'm happy with an FDR of 0.2, then I would need to remove, in this case, all the identifications that are below score 60, right? Like this is where I put the threshold. But then because the FDR is estimated with the decoys, the targets and the decoys of the database, then the estimation of this false discovery rate and therefore how I filter peptide spectrum matches depend on the database that I'm using in the first place. So we'll see how that will actually widely change how many spectra I consider identified. So basically, what we have here is for every database search software that we tested, for every search space configuration of database and of triptych or semi-triptych and specific search, then we have how many PSMs are above different FDR thresholds. And this is how we all work, right? We set a low FDR of, let's say, 0.05 or 0.01, and then only accept PSMs that are below. And as we have seen, the configuration of the search space directly determines how many spectra I consider as identified and therefore usable to answer my questions and my hypothesis. But what we see here is that always, in every case, the smaller dairy database always gives more identifications, even though the bigger one contains it inside. Also, non-specific search only gives more identifications when the proteins are actually degraded. So it wouldn't make in this case so much sense to use unspecific search for the almost non-degraded sample. And then the third thing is that all the open search software gives more identifications with the right set of parameters, of course. So this is not so much like, oh, this is better than that, or this software or that, those parameters are better than that. But this is just so that we think of how we operate and how we can get like widely different results if we use the software in different ways. But so what is happening here with the two different databases? This is something that we should think about. So what's happening is that increasing the database, in this case using the bovine proteome that has sequences, a lot of sequences that are not in the sample, that increases the chances of false, random target and decoy matches, which decreases the significance of the true matches. So let's look at this simplified example. I have the same list of PSMs as before from a search with a small database. Then let's say I do another search with a larger database with extra sequences that are not in my sample. Then boom, a decoy, because we have a lot more decoys. By chance, a decoy makes its way to the top of the list. Now to keep the same FDR threshold, then we need a higher scoring threshold because now we have more decoys that are accepted. So I need to move my threshold up. And then by doing that, what happens is that we end up rejecting matches that maybe before passed the threshold by using a larger database. So what's happening here is that because we're using this very large database, the assumptions underneath the estimation of the FDR are getting weaker. And then the FDR estimation is not working so well anymore. This even led to the idea that if I want a proper FDR estimation and for the purposes of significance, then I should not just restrict the database to what there might be in the sample, but to what I actually care about within it. And we sometimes do that, right? For example, if we use a collagen exclusive database to search data coming from bones, for example, even though there might be other non-collagenous proteins in the sample. Here we would be focusing on what we care about, not what there might be in the sample, but we don't care. So this is, this is something that we do and consider, but then what about contaminants? They are in the sample, but we don't care about them. Do we include them in the search or not? So I think, I mean, it's a good read, this paper, but we also need to think about the contaminants and whether we actually, it might be that we actually care about them, so we want to include them in the search. Let's also talk about open search. We saw that it increased the number of identifications a lot. So we know that ancient proteins are plagued with post-translational modifications, some that we account for and search for, but some others that we don't. And in this very simple data set, I found using MSFragger that a high percent of PSMs have other modifications than those that we initially and that we usually set. So this would be peptides with these modifications that if I use a narrow search, I probably would never identify. So what OpenSearch does is to allow a wide precursor mass shift between the observed mass of the peptide and the calculated one from the sequence, as you can see here. So this would be observed mass coming from the peptide, and this would be the calculated mass from the PSM between this spectra and this peptide. So they diverge, and let's say they diverge by this much. Then we say that we've identified this PTM, which is an exos, that maybe there would be some shifts that are completely unknown. We don't know what they are, but some of them do. And then this way we can discover novel modifications and account for a lot more. So what's happening here is that we are actually increasing the search space, right? Because now we allow a lot of modifications, but we are expanding it in the right way. So this has two effects. One is that what I said is we identify more spectra from peptides that have unaccounted PTMs, but the other is that we might be limiting the false positives, because in narrow search, by being too restrictive with the PTMs, the spectra of modified peptides would need to conform to a very limited set of options. And then we might end up matching a modified peptide to a different unmodified sequence, and this might be wrong. And so actually studies looking at previously unidentified spectra and clustering them found that a lot of those spectra clustering in groups with unaccounted PTMs. Let's move now completely different into de novo sequence. So what I did for the de novo sequence is I got my de novo peptides, and because the ground truth, the sequence that I know they're coming from, the BLG, is very simple. So what I could do is sort of using a blast-like method, map them all back to the actual right sequence. First, basically, we match N and C terminal k-mers that serve as seeds to extend the alignment, allowing mismatches and gaps. And this way we can spot position-specific errors between the de novo sequence peptides and the actual protein they're coming from. So we run 3 different de novo programs, and we also use within them the two different databases as before, either because some of these methods are actually hybrid and they need a database for extending de novo tags that they create, or also because they use, like at the end, they use BLAST for validating and reporting the sequences. So looking at the overall accuracy of each methods, what I found is that the accuracy is high for methods that depend on a database. to spot errors and inaccuracies, but it only works when we use the right database. So this is like a double-edged blade. We have a de novo method that can work and give us the sequences, and with the right database, I can discard and limit errors. But at the same time, a large database that has too many sequences that are not in the sample that we use for blasting, we can end up picking up errors that were initially committed by the de novo tool. So we can see here how the accuracy, the proportion of exact peptides always decreases if we use the larger database, so in all samples. Same would be here with the overall accuracy. You can see these distributions like sort of shift down if I move to the larger database. And another thing that we saw is that the N-terminal of the peptides is always less accurate. And that's because Y ions are preferentially generated in HCD with respect to the B ions. So because we don't have enough good B ions, then when the peptide is long enough, we make more errors in the N terminal. So now we can look at how these errors distribute along the BLG. So what we have here is like each amino acid instance across the BLG. So this would be the true BLG sequence. And then the matches are counting to the positive side of the x-axis, and then the mismatches or errors count towards the negative side of the axis. So in this case, for the tool Novor, which doesn't seem to depend much on a database to swipe errors, this is very similar whatever database we use. We have areas of the sequence in which errors accumulate as the degradation proceeds, and areas in which errors are more limited. So we have, for example, this area in which errors can accumulate. But also what happens, and this is important, is that we have areas with like a very decent coverage, like in the mildly degraded protein. But these areas with good coverage, they also have and accumulate errors. This doesn't happen, for example, with PepNovo, because PepNovo can use a database to identify these errors by mapping them back. So if we use the small database with just dairy sequences, then yeah, we get a lot of positive, like good matches and no errors. What happens if we use the very large database? Then errors start accumulating roughly in the same areas as we saw them with the other tool, with Novor. So PepNovo here is making mistakes in the sequencing, and then because we are blasting against a huge database, then something is blasting or giving like a good result, and then it's picking up those errors. But then we can also look at the data in this way, like plotting along like the coverage along the sequence, but not only with the de novo tools. So this is, for example, the same plot with the results with pFind using semi-triptych search and the dairy database. So here I want you to know to focus on something is how some regions are more reported, like this one, only after extensive degradation. This is because maybe it's only here that this region of the sequence is accessible to trypsin, or maybe it might be because this part of the sequence is more stable and therefore stands out when the rest of the sequence start disappearing. But then there are other regions that disappear, like there are many others, but I want you to look at this one. And I want you to look at this one because this peptide here that I'm marking green is a very common peptide that is very persistent in the archaeological record, and that has been hypothesized to survive well because of its interaction with mineral surfaces. Well, here it's happening the opposite. There's no mineral surface, and then after extensive like degradation, we don't pick it up so well. So this might be because it's not so stable, or it might be because at least it's not accessible to our methods, or it's not picked up, or is shaded by other peptides being more like prominent. So all in all, the conclusions, and I hope I at least gave you like more things to think about when you start running your analysis. But some ideas that I would want is to use, always want to try to constrain the search space that you are using when you use database search. This doesn't mean that you only look for what you are interested in the sample, but there might be things that totally like don't make sense for you to search for. In this case, it didn't make sense to look for the whole bovine proteome, right? Because I know that I will only get milk proteins. And in this case, even the smaller database, which contains dairy proteins, it contains dairy proteins from other species, not only Bos taurus. So again, maybe I can think about removing those, but also be careful because you cannot have like just very few sequences in your database, because that also complicates the estimation of the false discovery rate. But yeah, think about that, and also how the context of your sample interplays with this, and that can also give you hints about like what you want to search for. And I think you should at least give a try to the open search methods like pfind or msfragger. They are fairly simple to use, and I think they're gonna get you more results. Of course, as always, you need to at some point like manually inspect the data and make sure that the identifications are correct, but give a go to open search. And a common direction of the whole thing is sort of let the data speak for itself rather than search for and make it conform to a fixed set of things. But then what happens is that, yeah, de novo points in this direction, right? You let the data come up like however it wants to be. It will just give you the peptides de novo, right? But the problem is that what happens with errors that might go unaccounted, and what happens with the de novo software is still dependent on the database. So finally, how about integrating these different types of methods like narrow search with open search to discover modifications, and also de novo to discover like novel peptides, but also to start to have a first idea of what we have in the sample and being able to constrain our database. So that would be all from me. I want to thank. I would like to thank you everyone for attending the presentation, and also the people that helped me with all these analysis, and to yeah, to gave me a lot of things to think about with the results that I got. So yeah, thank you very much. And if you have questions, I'll be very happy to answer.
Thank you so much. That's amazing. It's such an incredible data set. So much data. I can't imagine going through all of it. And I think it's really interesting, some of the results you've had. If anyone has any questions, they're welcome to unmute and ask, or post them in the chat and I can dictate them to our speaker. But yeah, anyone have any thoughts?
I can start with a very random first question.
Mhm.
You said in the beginning that you're not going to recommend any best software to use, but do you have the opposite? Did you find a software or pipeline or something that is just really bad, that is very user unfriendly or has bad output or something like that, and you would just recommend that nobody ever touches it?
Not really, because I mean, there might be cases where some of them just doesn't work well. I mean, in this case, in this analysis, for example, with MaxQuant, I didn't get so many results, but there are also cases in which you get enough good stuff to continue. Also, I didn't even show it because I got even still less results. I tried AlphaPep, for example, and didn't work. And I think as of now, AlphaPep still doesn't have a semi-triptych search included. But so yeah, I think I don't think there's a method that or a software that I wouldn't recommend, but there are because you also have to think that I got the results that I got in this for this data set for this case specifically. So things might have worked slightly different with other data.
Thank you. And thank you for the great talk as well.
Thank you.
I'll jump in with a question. Thank you. It is fantastic work, and you've actually answered a lot of the questions that I had going into it. So I just started working in ancient proteomics, and I had a lot of questions. You just answered a lot of them, so thank you. I wanted to ask a very technical question. When you had the figures showing how much of the spectra you're covering, what is the 100%? Like, what is the maximum you can reach? Is that all of the MS2 spectra? Is that all of the peaks in the MS1? Like, what's the gold standard?
Okay, just a second. Let me wait. No, I need to share the other screen so I can move it up. Sorry. Okay. You mean in this plot?
Yes, exactly. So yeah, in the previous one, what was it, 100%?
You mean in these ones?
No, in the one you were on for.
Yeah. Here, right?
Yes, exactly. So what is the what is the 1.0?
Okay, yeah, sorry. So that would be all of the spectra that you have in your raw files.
So are those like the MS2 spectra, the
MS2,
secondary?
all the MS2s, yeah, all the MS2
Okay.
spectra, actually.
So if you're doing, so it also depends on the method in a sense. So if you're doing top 5 or top 10, you would have a different goal to reach.
Yeah, but I'm also picking up the best peptide spectrum match for each spectra.
Oh, I see. Okay.
So I'm doing like, I think if that makes sense, I'm doing top 1.
Fair enough. Okay, that answers my question. Thank you.
Maybe it's my time. Okay, well, sorry. No.
Oh, you can go ahead, sorry.
Yeah? Okay, perfect. Thanks. So, well, first of all, thanks, Ismael. It was amazing talk and amazing paper. And I've been reading the preprint, and I have some questions about it. And the first of all is about MaxQuant, because you mentioned in the paper that there's a way of making MaxQuant an open software, let's say. So have you tried, or I believe that this is done increasing the mass tolerance of the mass tolerance parameter. So have you tried? And
no, I've never tried it. What what I think I mean there that is that the open search is the most basic, is just that, is allowing that precursor white tolerance. So that basically you can turn every method into open search. But I've never tried it, and the reason for it is, one is that it might take a very long time, like software like MS Fragger that uses the same open search. It had to implement like a very sophisticated search algorithm to be able to cope with how you massively extend the search space. Also, pFine does like a more sophisticated search to again be able to use a wide precursor tolerance. And then the good methods like MS Fragger that are, I used it within another tool that has like a whole pipeline that is called FragPipe. Also has other tools that let you analyze the results that you get from the open search. So that's why in this case, if you want to do open search, you might want to go to one that sort of specializes in it, because it will also give you other tools to find it. Because if you do open search with Max1, it's just gonna give you, when it finishes, it's just gonna give you the mass shifts, but then it will be up to you to look for what they are. But pFine or MS Fragger in FragPipe, they actually go to a database called Unimod. It's a database of post-translational modifications, and they search for the mass shifts, and then they also report you back what the mass shifts in your data are. Like, is it from a glycation? Is it from anything? Like, is it it even has like things that are artifacts from like procedures in the laboratory and stuff like that.
Okay, thanks a lot. Regarding to this, I'm also concerned about the fact that when we are trying to do like paleoproteomics with really ancient samples, there are also mass shiftings due to SAPs like polymorphisms and so on, and substitutions that you're not taking into account. So also, I believe that this open software will also, let's say, allow us to identify this spectrum, no? Isn't it?
Exactly.
Because and also like the novel software is also meant to use to this purpose, but you also talk about that some de novo software that the most accurate ones also correct using a database. So that correction, do you believe that will also mask the substitutions, these SIPs that we are trying to always to find to build phylogenies and so on? Or is it like, I know that this is not the goal of your paper,
No,
but
but I get it. But yeah, so because you also have this Unimod database of modifications, it also sort of counts these mutations as if they were a modification. So you would be also able to see them and treat them as such and get them reported. The thing as with de novo and other tools is that you will have, if you have discovered a novel peptide from a novel protein, you at the end will also have to then go to the spectra and check that, manually, that it is what is telling you. It's not gonna give you, it's not gonna, in that sense, it's not gonna make your life easier because you also need to validate it, but yeah, it will be able to find those.
okay, thanks so much.
Hi, Ismael. Thank you so much for the talk. I just have one question. You said the narrow better for the database, and if I'm interested in, like, for example, if I'm interested in collagen, but I also want to see what other proteins are there, like how narrow is the database? Should I separate them into two runs, like one for collagen specifically and another one with the other like whole proteome database, or like one whole proteome database is narrow enough. And yeah,
okay.
sorry.
No, finish your question. I thought you had finished.
And I heard of another kind of practice is that for certain animals, the sequence is not that complete, and so people add a more complete but similar animals database, for example, BOSS, into that database, and to capture those are not counted by the origin the animal that we are interested. So that certainly increase the database. And like, how do you think about this?
Yeah, I mean, this is one of the reasons why there's no, I mean, there's not this like thing of, yeah, make your database small enough. It's not an absolute thing, because you definitely have cases like that you might have like extinct species or species that are not well sequenced, and then you need to include other like homologous proteins from other species to try to get those peptides that might be different. This is also in part where open search might help you, as we were talking before. So yeah, I mean, this is one of the reasons why there's no like too much right or wrong. And then regarding the collagen, I mean, it depends on a bit on like what you want to focus and what you are looking for or trying to answer. You might want to try, why not, two different searches. One with like, if not the whole proteome, but like bone related proteins, and then another one just with collagen if you want to focus on that. I mean, that would be, as I said, like up to your research question.
Okay, thank you so much.
I have a question, actually. In terms of MS Fragger, I know that there's the option to add the decoy database, which doubles your database. And so if you're trying to use SwitchProt, I've never gotten it to really behave. And I know SwitchProt, like you were saying, it makes your search space huge, and I'd rather not use it. But so, because I really like MS Fragger and I would like to use it, but I know that they have that option to split the database into many segments, essentially, to kind of make it easier on the program. So have you tried that, and has it altered your data in negative ways, or is it?
That that shouldn't change. That shouldn't change your results. What it's doing is just instead of getting the data and then for all of it like breaking it into peptides and then generating the in silico spectra for all of it at the same time and then doing the search, it just does it little by little. Once it has done everything, it will collect all the PSMs and then give you at the end like the FDR and the statistics. But that part is done already with all the database already like searched through.
Amazing, thank you.
That's just because, and I actually had to do it because I ran into that problem when I was running the whole bovine proteome with unspecific. Then like it would just overflow the memory of not just my computer but the cluster at the university that I was using.
Yeah, how many databases did you end up splitting it up for the non-specific? I'm just curious.
I can't remember in how much I split it.
That's fair. Thank you.
No worries.
We have about 5 minutes left. So any final questions for our speaker? Comments, concerns, feelings? Personally, I'm feeling a little like I need to go and fix all my parameters for all my searches. I do have one last question, actually, because I'm curious about this missed cleavage that you mentioned in the last slide, which I'm assuming you're picking up from this semi-triptych search. Yeah, you're accounting for the missed cleavages in some of your samples. So I have been doing these extraction experiments, and I'm finding that some of the extraction methods are more likely to have missed cleavage in the data set versus the other extraction method. And so obviously one of them is turning out to be better because it's having these longer peptides, which are more specific. And I'm wondering how much of that, I mean, I'm running them both through MaxQuant. They're just going through SwissProt, MaxQuant. It's just for the comparison of the performance of the extraction method. But I'm wondering how much of that is actually probably MaxQuant in its processing of that data, and how much of it is actually the extraction. And so I thought I'd ask.
Yeah, I don't know, because the miscleavages of like and how we use the trypsin is not something that I looked like thoroughly through this data, but it's something that also like Matthew and I are interested in looking at, like where, especially in collagen, for example, like where miscleavages would tend to happen more often than not. But I don't know to what extent that's also dependent on the software, because the trend that I saw, for example, that's part of the sequence that disappears and others that come up after the extensive degradation. That happens like for most of them.
All right, interesting. Thank you. Matthew, you want to say something?
I want to ask Ismael a question, which I don't know the answer, but maybe he does. I mean, obviously, you've been looking at all these different software tools, and they do things differently. Have you got any sense about the different strategies they use to identify the MS2, which should intrinsically make one more better than another for ancient samples?
Well, I mean, if we're talking about like ancient samples,
I mean, the kind of stuff interesting in this community, you know, in the sense that the different software tools have adopted different strategies, and we kind of tend to think of them as just packages we plug in and run. But I was just wondering what, you know, obviously you've looked at all of them now, and we work on degraded proteins and more damage. I just wondered whether you feel that some things, the strategies that people have adopted in the software are actually more attuned to the kind of samples we have.
then I mean, I think I would say software like pFine and FragPipe that has MS Fragger in it are because how they report the data, because especially FragPipe, you can also sort of customize a little bit your pipeline, but then especially because they're open search, I think there are methods that make a lot of sense for us. Because I mean, we have a lot of unaccounted modifications that are unaccounted for, especially with ancient proteins. And you could see here only like with this very simple data that there were already things like going on.
Thanks.
Okay. Well, if there's no final questions, then I think we'll wrap it up there. If anyone has any further questions, I'm sure Ismael would be happy to respond to an email, or maybe this article is in preprint and he doesn't want to have to think about it ever again. But regardless, that'll be the end of this webinar, and we'll see you all next month. Thank you so much for the amazing talk, and thank you everyone for coming.
Thank you, Lindsay, and thank you everyone.
Bye, everyone.